So hello all, and thank you for attending the Launch Bio Design for Success Early Phase Study Design for Biotech. And we have speakers, Caroline Engel, who's from Celerion, and David Warchuk from Brin Pharma. And I may have messed up his last name, so David, I will have you correct that. So I know these past couple months have been difficult, so we really do appreciate you choosing to spend time with us today. And my name is Natasha Eldridge. I am the program director for Launch Bio in Cambridge and Boston. And Launch Bio is a nonprofit organization with a mission to build on the ecosystem of innovation for the benefit of human health and well-being. For more information on upcoming webinars, including the Compensation Confidential we have on April 28th, we will be going in depth in life science compensations for 2020, how COVID-19 has impacted salaries, lab operations, and even succession planning. You can find all this information and more on our website, launchbio.org. And through the help of our sponsors like Celerion, Launch Bio is able to provide these webinars. So if you're interested in becoming a sponsor or would like to see who is currently a sponsor, you can also go to launchbio.org to check us out. I am excited for our program today on clinical study design and our guest speakers. First up, we'll have Caroline, who is the principal scientist of protocol design and development in clinical pharmacology at Celerion, where she has actually worked for the past 19 years, and 16 of those have been in the protocol scientist department. She has worked on studies from early phase 1 to phase 2 proof of concepts. And then following Caroline, we'll have David, who is the CEO of Brin Pharma. And David has more than 35 years of experience in pharma, bio, molecular diagnostics, medical device, and the CRO industry. Prior to joining Brin Pharma, he directed the Life and Health Sciences strategic development effort at Oracle and also served as a vice president at Quintiles. So don't forget to ask your questions via the Q&A app at the bottom of your screen, and we will leave the last 15 to 10 minutes to answer your questions. So Caroline, I will let you take it away.
Thank you, Natasha, for this introduction. So I want to start with saying to all of you, congratulations if you are interested in early phase 1 studies, because you have potentially selected a molecule as your main candidate and move on to your next milestone. So today, today I really scratch on the surface on how to transition into early phase 1 study, what essential information and data you need to design those studies, what option to consider for study designs, and how to select your CRO for an efficient partnership and facilitate this transition into your phase 1 study. So if it's not already done, I strongly suggest to read upon the guidance provided by the regulatory agencies on the subjects. These are a few examples for the FDA. Even if you're not planning to submit in Europe, I also suggest to read upon the European guidance. I think together with the FDA guidance, they provide an overall well-rounded picture on what is needed for these types of studies. Unfortunately, we're going through tough time with the pandemic right now, and it would be useful as well to familiarize yourself with the guidance on the conduct on clinical trials during the pandemic. So what is the main objective of the early phase 1 study? The main objective of the early phase 1 studies are mainly safety, tolerability, and pharmacokinetics. You might consider looking into pharmacodynamic and special consideration to assess some extrinsic and intrinsic factors of your drug. These studies are typically performed in a healthy subject population because you want to really target the safety tolerability of your drug and not have the comorbidity of your patient influence this profile. However, some drugs unfortunately are toxic and cannot be administered in this population, and therefore you have to look into administer it into your patient population. So what scientific resource do we need? As a scientist, it is important for us to give you educated suggestion and feedbacks to provide you appropriate designs, efficient designs for you for your study to be properly performed. And that resource, we need it from you, mainly from the investigational brochure. Unfortunately, sometimes in early phase 1, this document is not available. It's in the draft process, so a pre-IND package, if you were planning to meet the FDA, will be appropriate. And of course, if this is not available, then we will need a non-clinical study report. Mainly what we need as a minimum is the 14-day or 28-day toxicity study that were used to establish the NOAEL, information on the toxicokinetic of the drug, animal data model that was used to establish your pharmacological active dose, and if possible, a dose response curve. As an extra, sometimes it's not always available, but I appreciated absorption, distribution, metabolism, and elimination of your drug in your animal model is very useful. This data helps us well design your studies and calculate your safe starting dose for your first in human. I want to point out that the FDA guidance on establishing the safe starting dose clearly indicates that investigator involved in these clinical trials has to agree on how this NOAEL was established. And sometimes the clinical significance of some clinical observation may bring discussion. So it's important to have early discussion to really understand how you establish that NOAEL and what's your scientific rationale for us. So it helps us in the process and make it more efficient. So how do we calculate that for that safe starting dose for your first in human? The traditional route is a stepwise approach where the NOAEL was determined in at least 2 animal species, a non-rodent and a rodent species. Based on this, we converted using an allometric scale to human equivalent dose. Based on this information and from your data, we select the appropriate animal species. We apply a safety factor, which gives us the maximum recommended starting dose. However, is this the most appropriate approach for your drug? Based on the data you're going to provide us, we need to take into consideration other aspects. Is what is the dose response of your drug? Was the toxicity? Was the safety factor appropriately selected? And is the animal model is really relevant? Is it a good model for a correlation into humans? And most important, how did you, what's the pharmacologically active dose that was selected? Is it lower than the maximum recommended starting dose? Therefore, we may adjust these doses accordingly to provide a safe starting dose, to provide a dose that the initial dose will provide no toxic effect to our human patient, and we well characterize your therapeutic range in this study. This is a traditional first-in-human study. In this design, we simply assessing single dose that is performed in an escalation manner in a limited amount of subjects. Currently, the FDA doesn't have any written recommendation on sentinel dosing. It's left at the discretion of the sponsor. As scientists, based on the safety profile of your drug and the nature of your drug, we might recommend to you to administer it to perform sentinel dosing to make sure the safety of the subject are appropriate. FDA is known sometimes during the pre-IND meetings and IND submission to ask to add sentinel dosing. So it's not because it's not recommended in any of the guidance that they may not require it for your specific drug. For European guidance, sentinel dosing is required at all dose level in the single ascending dose and in your multiple ascending dose. What we also would recommend, if it's an oral dose, select a cohort that may cross over into fed treatment to assess the effect of food on your product. In most recent years, we've seen an increase in evaluation of cardiodynamic ECGs in your first in human studies. We have to realize that first in human study design is the it's the rare opportunity that we have to characterize your drug at different dose levels. So having intensive ECG monitoring in this type of design provides you a well characterization of your cardiac safety of your drug. And it's valuable data that's going to help you in the future to take a decision if you want to continue with the drug development, how to change your monitoring in the future studies, and potentially if you have reached high enough exposure that meets the regulatory agency requirements, you may take this data and request for a TQT study waiver. So this design is a typical simple first-in-human design. This is performed in the clinic within 6 to 8 weeks, and not long after, you have in your hand a clinical study report and initial information on your drug to either meet regulatory agencies or investors that may help you take decision for your future development of your drug. However, if you look to gain more information on your drug in a shorter amount of time, then we would suggest to add a different part to your study. You can add your multiple ascending dose to your first in human. Of course, this is performed following safety tolerability review and interim PK analysis to help us decide on dose levels and regimen you want to use for this portion. If it's feasible in healthy volunteers, you may want to add PD and biomarkers. And as well, you can consider adding additional cohort that you want to evaluate extrinsic and intrinsic factor of your drug. In addition, if you want to push the envelope a little bit more, and you want to gain early information on the possible effectiveness of your drug, you can potentially add 1 or 2 cohort in patient. This also helps if for some reason the relevance of the data in healthy subject may not necessarily correlate in your patient population. For example, cystic fibrosis patients are well known to have a different way of absorbing nutrients and drug. So it would be advantageous to add this in your first in human to compare what's the difference in PK and safety profile between your healthy population and your patient cohorts. So this provides you a wide range of information for your drug product. However, you have to take into consideration that clinical study report will have to wait until every portion of the study has been completed. So during the pandemic, there is, of course, an impact on our study design, but mostly on our clinical conduct. I'm not going to go into details because I have a colleague, Arnaud, who's going to be presenting on May 19 on clinical operation and the effect of this pandemic. So I suggest to tune in to that Launch Bio webinar. So in general, of course, the clinical conduct of our study will be affected. The clinical flow will have to change to prevent potential cross-contamination. As scientists, we like to have as much as information as possible from our clinical trial. However, we have to take into consideration right now what is really clinically relevant and essential compared to nice to have. So we need because we need to manipulate our subject as less as possible, so it needs to be taken into consideration. So the confinement will be possible, but of course in smaller groups and control environment. We will need to consider the epidemiology of the virus in the surrounding neighborhood of the clinical site to make sure that we don't have a high risk of contamination. We will control access to the clinical sites by screening the trial subjects and clinical staff for the virus, but as well for flu symptoms like body temperature on a regular basis throughout the study. So currently our bioanalytical lab is validating a PCR assay to measure virus in our subjects and our staff, as well as an antibody assay. So this will help screen this situation. The silver lining in all this is it pushes us as scientists to think outside the box and find opportunity to innovate, such as implementation of virtual assessment and home visit to replace our traditional site visits. So what to look into your CRO when you want to select that partnership. These are the key elements I would suggest to look into. So what's the CRO expertise and experience specific to your design? So for example, and if you need help in regulatory regulatory specialists that can help you meet the FDA, answer some of their questions. Do they have that staff available by the CRO? As well, do they have the scientists and clinical staff to conduct your trial and to analyze your data at the end? And what's the experience do they have in recent years similar to your design? Their facility and equipment, do they have the proper clinical site facility and laboratory to perform your study? If you are planning specific PD assessments or you are planning a phase 0 trial with a radiolabel agent, do they have the capability to do it? Do they have a clean room, nuclear license? Do they have the capability to perform those assessments that you require for your study? You're planning pulmonary function tests or echocardiogram or hemodynamics. Can they perform this? If you had cardiodynamic ECGs on your study, do they have an ECG core lab on site to be able to quickly assess those data and analyze it for you? And what's their access to volunteer and patient? Do they have a good database in-house, or do they have partnership with external sites to recruit and enroll those panels that you need? And last but not least, what's their regulatory observation in recent years? And really, how did they respond to them, and how they improve their statement, their procedures? So these are, this ends my presentation. I want to say thank you to Launch Bio, to all my colleagues at Celerion that helped me with this presentation, but on my everyday work from which I learn every day, and our sponsors that always provide innovative way to treat our patients. And one of them is David.
Okay, so thank you all for joining the call this afternoon. I'm sure most of you have nothing else to do but sit out in your home office and hopefully be productive. So I'm going to go through a little bit of what we're doing within Bruin Pharma. But just as a starting point, just to give you a little background, we're a virtual startup company. We've been around about 3 years. We have a massive number of people within the organization. It's a total of 5. My 2 partners and I started the company and really tried to take advantage of a situation or in a clear patient and medical need to build an alternative to be able to help treat a number of patients. So what we're essentially doing, and I don't know how many of you on the phone are familiar with or have a direct patient exposure to or directly afflicted with some sort of severe allergy, perhaps a food allergy or bee stings or latex or drug or whatever, that results in an anaphylactic or could result in an anaphylactic event. The most common therapy now is epinephrine, and epinephrine is not a new molecule, as you all know. And so other than what Carolyn was talking about and all these cool things to do with a new chemical entity, we didn't have to do any of that because epinephrine's altered in dirt. People know what it is, know what's happening and how it works, and what people are trying to effectively do with it. The current therapy, if you will, is all through some sort of an injection, either through some kind of an auto-injector, primarily the most common one being the EpiPen, or if you do end up in an emergency room with some sort of either infusion or an IM injection also of epinephrine in an attempt to get your symptoms under control. But the whole thing is that for the most part, patients are not adequately protected. And just give you some demographics of what's going on in the market space, you get a sort of feel for what's going on. Give or take somewhere around between 40 and 50 million patients are at risk for anaphylaxis. About 16 to 30 million, depending on which epidemiological study you look at, are in fact diagnosed with anaphylaxis in some way, shape, or form. Generally, food is the most common kind of scenario. About half of those patients actually get a prescription for some sort of epinephrine auto-injector in some way, shape, or form. And only about half of those actually carry a device, and about 5% of those who carry a device carry 2, which is the recommended event. Because in about 20 to 30% of the cases, when you do have an anaphylactic event, roughly 2 to 20 to 30% of the time, you'll need a second device or second dose. And while you're trying to get to emergency medical attention or control symptoms. Anaphylaxis is of a strange kind of a situation where it gets worse and worse and worse as time goes on, to a point in time where if it's not treated effectively, it unfortunately results in death. And again, depending on what epidemiological data you look at and how you look at it, it's anywhere between 1,000 to 1,500 patients a year die of anaphylaxis, which is really, when you think about it, quite strange when there is a known proven treatment for anaphylaxis. However, patients simply don't carry the devices or have access to treatment. So it's an underdiagnosed and treatable event if you have the opportunity to do so, and hopefully people do. So one of the things that we did, and I'll show you a little bit about what we were looking at as an alternative, but we looked at a patient preference study of about 180 subjects, and we asked them saying, if you had a nasal delivery device for epinephrine versus auto-injectors, what would you prefer? All right, and so we looked at it in the perspective of a number of key parameters upon which the subjects or patients talked about what was interesting to them or important to them. Portability being one, ease of administration. The perceived safety issue was also quite interesting, and the size, which essentially goes the same way back to the potability problem. Because the epinephrine auto-injectors, and if for the folks who carry them, they're quite big, make it extremely difficult for people to carry them around, especially if you think about an 8, 10, 12-year-old kid. Where do they put them? You know, boys don't want to put them in their pockets because they're too big. You know, girls don't want to carry them in their purses and their backpacks because they're too big. And interestingly, because of a needle, many schools won't even allow you to have them in the building because they have needles and can be perceived to be used as weapons. So it's quite a problem when you really think about it from the patient perspective or mom perspective. When we talk to physicians, this is a little bit of a different story, but still similar kind of thing where portability was being a key issue. As in talking with numerous allergists, what we found is that the number one thing that keeps them up at night for these patient populations is that simply the patients don't have the device with them and are not treating themselves appropriately when needed to. So it's a big problem, and the biggest issue is because they simply don't have it with them. Interestingly, we just completed another study. We termed it as time to treat. So how long does it take you from onset of symptoms to dosing yourself or a patient in your care? And the overall, we've seen anywhere from 10 minutes to 90 minutes from onset of symptoms to delivering epinephrine. And when the symptoms do get worse over time, obviously if you're waiting 60, 90 minutes, the probability of effective control is pretty low. But we also found out that 42% of the patients, which is consistent with some of the literature we've had, didn't have the device with them at the time of an event. So again, very problematic for the effective care treatment of these patients. So what we've done is essentially build a novel bi-dose nasal delivery device, which is shown in the middle of this chart. So on the left, you can see the auto injectors, essentially in this case, the EpiPens. And they have a clip with them, and that clip is there on a purpose. You're supposed to carry both of them. So if you look at this as a size comparison, and I'm sure there's going to be some lawyer someplace saying I shouldn't have an actual product in here, like the chapstick, but it gives you a good perspective of size of the device in its carrying case. And then you can see in the pictures on the right, it's small enough for, in this case, a 12-year-old girl to put it in her pocket, or a little clutch purse in that scenario. So it's easy to carry with them, which is one of the biggest issues of portability. And being a nasal delivery device, there's no needle, so it doesn't hurt. And I am anaphylactic, and I have done the auto injection, and it does hurt. But this case, the whole device itself is non-intimidating, it's easy to use, and there's no needle. So hopefully addressing some of the key issues of portability, ease of use, and reluctance to use something with sharps or needles. So that's sort of a background of how we got to the product. So some of the things that Carolyn was talking about in regards to what you need to do to get into the phase 1 or first-in-man studies, we didn't have to do, simply because, again, we're working with a proven known entity of epinephrine. But then the complicating thing is that it's not just a simple chemical entity as a drug. It is, in fact, delivered with a device as a combination product. So unfortunately, or fortunately, I'm not sure which way you want to look at it, we don't have one overseer, we have 4. So we have the Bureau of Drugs that we have to worry about, obviously because epinephrine is a drug. And then we have devices. So that part of the FDA is directly concerned with the device itself and everything surrounding its complication, its manufacturer, its quality, et cetera, et cetera. And then we have the combination product group. And this group is really looking at the perspective, and interestingly, which has become more and more important over the past couple of 3 years, is reliability. Is essentially how reliable is the device to deliver the drug at the dose it's supposed to do almost 100% of the time. So it's referring to 4 nines and 5 nines, so 99.99% or 99.999% reliable for the different aspects of effective drug delivery through that combination product or the drug device tool set. These require a whole separate set of programs and activities and assessments that all have to be done in parallel to getting to registration. And then we have the other key piece is the human factor side, which is when you actually give the device to people, how do they use it? And can you give them instructions and training to use the device appropriately, which is one thing. And if you're thinking about it as sort of an inhaler or nebulizer for asthma or something COPD, is one thing where it's a relatively commonly used device. People use daily, weekly, whatever. In our case, the average times that people use these devices is 3 to 5 times in their lifetime. So the key issue here is that what happens if a lay passerby is the only person who's around to be able to give you effectively treat you with your device? How does that person do it? And can they do it and do it effectively? So these are just other complications we have to deal with in the perspective of the development process. But in the first thing that we did, however, is we did go to Celerion for the issues and sort of the CRO criteria, selection criteria that Carolyn talked about. We did look at several CROs, and we centered on Celerion for a number of different reasons. The main ones were the ones that you described. But some of the key things there was in fact the facility, the expertise and capabilities of the people. And the key thing there was also familiarity with doing nasal delivery device testing. And this is not a trivial event in the schema of being able to do things and do it consistently and getting sort of commonality, if you will, of approach. But that was in selection of Celerion, that was part of the assessment modalities and methodologies that we did do. And fortunately, Celerion came out the top of the list to be able to run the studies with them. But some other things that we actually, once we chose Celerion to do the studies, was to really be thinking about how to do them. And then what are the factors and issues and concerns that we had to prospectively think about to effectively make sure that we're minimizing the level of variability or the issues that could in fact impact the outcome or the ability to interpret the data once the study is completed. And I just put a few of the things in here that we had to consider. And each one of these had to essentially be a separate, almost a complete process and procedure assessment methodology of what to effectively do and how to do it. To start with, epinephrine. All right, just by the nature of epinephrine as what it is, obviously it's an endogenous compound. We all have it, and it's made in the adrenal gland. It has a small depot. It's a fight or flight syndrome. And the issue there is when you need it in a bolus, but and the half-life, the endogenous half-life in circulation is about 3 to 5 minutes. So it comes and goes very fast, but when you have an allergic reaction, those symptoms go on for 10, 20, 30, 60 minutes, and therefore the epinephrine is not there because there is no bolus, it's a depot of epinephrine. Once it's gone, it's gone. It takes the body a long time to make it again. So therefore, emergency epinephrine is the treatment of choice. And in this particular instance, in these indications, epinephrine is in fact an emergency product. So when you think about it in the perspective of being able to assess and understand what's going on, you're not looking at it as a chronic therapy, as a daily therapy, weekly therapy, or even in some cases as a rescue, like for asthma, if you will. But truly is an emergency therapy. And if you think about it, similar to like Narcan for reversal of opioid overdose. It is an emergency situation and has to be treated quickly. Otherwise, there's significant sequelae that none of us really want to involve. So we had to be thinking about this as the stability of the epinephrine itself in plasma or in whole blood, and how to handle the samples. This particular instance, and we were, because of the short half-life of epinephrine, we were drawing samples over 6 hours, essentially 17 samples, 18 samples over 6 hours. And the vast majority of them were in the first 30 minutes. So thinking about how quickly you can draw a sample and then handle the sample, because the sample has to be kept cold almost as instantly from when it's obtained, and then spun down and frozen as quickly as possible after it's collected from the patient. So in these cases, we were handling and picking up the samples, taking them to the lab in the building, and having them from sample collection from the subject's essentially bedside to the minus 20 degree freezer in between around about 40 minutes. And if you're thinking about that, 12 minutes of that is the spin time. So, roughly 30 minutes from collection to freezer. So how to handle those samples, because each sample had to be spun down, essentially separated, frozen as rapidly as physically possible to ensure that there is very little degradation of the epinephrine within the sample itself. So the other key thing is that obviously epinephrine is an endogenous process, a product in your system, and it's a highly variable drug. So in the schema of the study that we did, just to give you a perspective, we had samples from patients of roughly, say, 20 to 100 picograms per mil to over 18,000 picograms per mil. So highly variable drug is to put it mildly, to try to understand that and control for the variability is a significant event. So one of the key things we wanted to wrestle with is how to define a solid baseline. When we did our animal studies, we found that we tried to draw 2 blood samples before we dosed the dogs. we found out that the variability was too great. So we ended up doing a 3 time point baseline of 30, 20, 10 minutes pre-dose, and then started dosing to try to get as best as possible essentially a basal level of epinephrine in this situation. So, but if you think about it, anybody in a clinic is going to have some level of anxiety, right? Because they're in a new environment, they're sitting in a hospital bed, there's people all around them, they're getting them ready to dose, there's timing, there's monitoring, there's all kinds of things going on. So it was some of the critical factors we had to keep in mind as to how to establish as calm as possible an environment to make it seem like this is no big deal. Because any kind of issue raises your epinephrine level, and you get your blood pressure up, your heart rate up. And those are some of the key factors for the dynamic effects of epinephrine. Primarily, it's heart rate and blood pressure. So you want to make sure that we're trying to control for that. But those are some key pharmacodynamic parameters that give you an indication of whether or not there's epinephrine in the blood system to begin with. So when you're looking at it at the pre-dose time, you have to be careful to establish a baseline and then monitor it essentially continuously during the course of post-dosing as a measure of the dynamic effect of epinephrine that correlate with the kinetic effect of epinephrine in systemic circulation. So trying to handle all these things was, let's say, put it mildly, not for the faint of heart. In the schema, when we would dose people, we would have roughly 10 to 15 subjects in a room, and with the different times, essentially places for dosing to collect the blood samples from them. But we had probably 6 people per subject to manage the control of heart rate, blood pressure monitoring, dosing, and taking out essentially giving the blood samples, and then handling the blood samples. So figure 5 to 6 people per subject in a room, obviously orchestrating that event and making sure, again, to try to keep it calm and rhythmic so it doesn't create anxiety into the subjects as they're being dosed and seeing what's going on. But also, we're also comparing against needle sticks with either an IM injection or an auto injector of use of epinephrine into the same crossover design. So all these things are going on at the same time to try to understand and control for the variability across subjects. So trying to understand how this thing really worked in the perspective of the actual procedures and working time for delivery to blood sample handling and for the 6 hours after dosing was quite an event to make sure to be thinking about. So it's one thing when you're like Carolyn was talking about, here's all the things you have to do to be able to get ready for the sampling and testing, to actually run the test, run the protocol, makes all the other stuff seem like a bureaucratic exercise. These things are the ones that actually, where the rubber meets the road, actually makes or breaks the outcome of the study, to be able to effectively understand how the variables are effectively controlled and be able to effectively dealt with, to be able to make sure that you have the most robust and high quality data at the end of the day from that study. So, and then that's only the drug, right? And then you have to be thinking about the device and the delivery. So how the nurse delivered the product, and making sure that the nurse who was delivering the product was trained properly. So luckily for, I don't know, good or bad for the Celerion team, we drew upon quote unquote volunteers from the Celerion staff to be subject to training, so the nurses could train on how to use and dose patients with the device that we gave them. So we gave water-filled devices for testing and training, and then also to try to assess what happens when you actually spray this into somebody's nose. So delivering the dose, one of the things even for screening criteria for the subjects, we used a delivery of just water into the person's nose to make sure that they didn't flinch or have a problem. Once we were doing the delivery, again, trying to control for that, there's another variable into the dose delivery process. So luckily we had one nurse who was excellent, do all the dosing, again, to try to minimize any differences. And then we also had to be looking at the actual performance of the device. Did it work? Sort of the reliability factor. So how do you do that? So in this particular case, because the device is essentially a closed system, the only way we can do that is actually weigh the device. So we had to have a balance in weighing the device before and after to 4 decimal places to make sure that we can actually measure how much of the dose was given, because we knew exactly, well, how the device is supposed to deliver, how much it's supposed to deliver, and to make sure that there is no huge variation of weight that would say we either had a high dose or a low dose in the overall sort of volume of delivery. So, luckily, I mean, we were able to handle all these things. But I guess to the folks on the phone, I would just strongly suggest that if you're thinking about doing these studies, or any kind of study for that matter, and looking at the capabilities and expertise of the team, think about how the actual logistics of the study itself are going to be delivered and how it's going to happen, and then work from there. Because those things will, in a lot of ways, make or break the study. So I'll stop there, and then we can open it up for questions.
All right, excellent. Thank you so much, David. And let's also bring Caroline back here. And so we do have a lot of questions going on. And so the first one I have is actually for you, Caroline. It's from John Sikowski, and he wrote in the Q&A. He says, is the process laid out here, particularly the SAD / MAD diagram slide, also applicable for large molecule P1 trials, or are there significant differences?
So for biologic, we would follow.
Oh, can you bring your voice forward? Sorry.
So for biologics and large molecule, we would follow the same trend. Of course, we will look into the safety of that molecule. Sometimes we can administer a single dose, but potentially not a multiple administration dose. We need to go in patient for that portion. So we need to look into really the characteristic of the drug.
Okay, thank you. And for you, David, we had Richard Zimsky. He wrote in a couple of questions, but I'll start with his second question, which is, at what point should we start engaging with the FDA? I know you just spoke about working with the FDA, so I just wanted to see what you had to say about that.
Yeah, well, let's just put it mildly. If you haven't already talked to the FDA, you're too late. So really try to get to the FDA as fast as possible. We started talking to the FDA in our preclinical program, even before we had the IND. We had some questions we were looking at, trying to understand. And rather than trying to figure out things for ourselves, we just put a question into the FDA, and they would answer and try to get something back to you, give you guidance. And when we then submitted our pre-IND meeting request, they were extremely responsive. They gave us an answer back in 3 days, which was unheard of for us. We expected 2 to 3 weeks, but they responded back in 3 days, and they told us exactly what they wanted us to do. So I would encourage you to talk to the FDA as frequently as possible, as soon as possible, but not just using them as a consulting group, really looking at answering specific questions. Because if you ask them, what should I do? They'll tell you, and in some cases you don't want to know, or you don't want to hear that. But if you ask them specifically, we want to do this, we want to do that, would you agree, yes or no? And often cases they will tell you yes, or sort of yes, but you need to add this thing, that thing, or change this or change that. And you give a very clear direction from them of what you'd like to do, so, or what they would like you to do. So I would highly encourage you to talk to the agency as frequently and as quickly as possible.
And to follow up with Richard's other question, and that was on any CNS-specific guidance or advice regarding regulatory process and trial design?
Yeah, the CNS guides, or anything that you think about as completely subjective assessment, which essentially like depression, OCD, any kind of schizophrenia, those kinds of things are very subjectively assessed. So in some cases, you really have to work hard at effectively doing the validation of your testing methodology before you start doing the actual testing to try to understand and assess efficacy or safety. Because it's so hard to grapple with, how do you feel today, or level of depression or anxiety, when it's all a subjective assessment. So in some cases, it's the rater and the rater variability, rater checking, or also what kind of online modalities you use, whether you're using some sort of ePro system or COA system, or any kind of a mechanism to effectively assess the subjective sort of relationship of what the patient is feeling or sensing is hard to do. And then you couple on top of that with the CNS scenarios, it generally takes 3 to 6 weeks before you start seeing any kind of an effect of any new moiety you're trying to introduce into that patient in their clinical setting. So there's a lot of variables to be considered in doing this stuff. So again, talking with the agency is early, and also get yourself a good scientific and clinical advisory board to really try to help you understand and design the protocol, what your inclusion exclusion criteria are, and your efficacy and safety criteria or your monitoring criteria, to really understand how you can get the best sort of outcomes, if you will, from those respective patient populations.
Excellent. And Caroline, this question is for you. There's been a couple of questions actually that have come in regards to COVID-19 and the number of clinical trials that have been postponed. And this is from Ted Benson. Have you already successfully applied Celerion's adapted clinical conduct concepts in trials?
To my knowledge, unfortunately, we are still working on it, on how to update these conducts and how to change our clinical trial methods. This is still in the works, and I think Arnold's presentation in May will be able to answer more of these questions.
Okay, yeah, so on May 19th, Celerion will be coming back to discuss clinical trials and what's happening with COVID-19. So I would love to see you guys register for that one as well. And then Caroline, I will end on this one with you. There was a couple questions also asking about cost. You know, what is the variable effective pricing for the type of work you're going to talk about, ballpark prices, estimations of cost and timeline for each phase. So I don't know if you want to talk about that or have people go to the website.
No, so I can have a brief information on what usually drives the cost is the sample size of your trials, the confinement of your subject in the trials, how long they're gonna stay in. And after that, there's also the type of procedures. Some procedure might increase the cost more than other if they're not the standard safety monitoring or PD assessments. So these are all driving factor that will influence your costs. There's a great article, if I can share my screen, if that's okay with you.
Okay.
So, okay, I'll go back here. And it's from Sertkaya 2016. This is freely available online, and it provides lots of good information on the costs for each of the therapeutic area at each of the phases. And the conclusion of the article clearly indicates that the driving factor is your sample size. So to take into consideration small molecule with simple safety monitoring, of course, will be easy to assess and doesn't require, if they're not variable, don't require a lot of subject on study. But if I take ophthalmology as an example, it's really hard to assess PK from an ophthalmologic product because it doesn't go into the systemic circulation. So you have to change your endpoint to a PD assessment potentially to compare, let's say, in your DDIs or food effect. So these PD endpoints are much more variable than a simple PK assessment. So therefore, it will drive your cost higher.
Okay, well, thank you so much for that. And thank you, David and Caroline, for your presentations today. I think they were very educational and informative, and I think our audience really did learn a lot from you too. So thank you for participating in this program. And for the audience, thank you guys again for attending. We know it's a super busy time for you and there's a lot happening. So for you to make time for us, we again truly appreciate it. Make sure you go to launchbio.org to see what our upcoming webinars are. Again, we'll be with Celerion again on May 19th to discuss the COVID-19 and the clinical trial situation. So we do have a couple of programs coming up next week, so check out our website, and I look forward to seeing you guys all soon. Have a good day. Bye.
Okay, Natasha.
Bye. Thank
you.
you.
Bye-bye.